might not be effective in preventing the transcription effect.
That's true. We don't actually know. You're right. Yeah. Although for the one the anti-CRISPRs all, like the first three characterized, all have different mechanisms for inhibiting cascade. So even if the one that was shown yesterday doesn't work for DCAS9, there's some rationale to think that we could find one that would work, but we'll see. We haven't tried.
So see, crabs not is that the best stand?
We've appended it to both, I think. No, I actually like the C-terminal fusions better. I think they're a bit more stable. The N-terminal fusions, you can see the protein is quite unstable, so when you wash out doxycycline here at day 4, by day 5, the protein by Western blot is almost completely gone. The half-life is quite short in human cells. That's a good thing for an inducible system because it makes it less leaky, but it means to get in our hands, you need a reasonably strong promoter to get full activity of these N-terminal fusions, where you can use a weaker promoter for the C-terminal fusions just because the protein's more stable. So the bottom plots also show that you can use this to study essential genes. So one of the things that is a little bit tricky to study using wild-type Cas9 is an essential gene. If you make an edit to the genome to knock out an essential gene, that's a bit difficult to recover a dead cell. With CRISPR-I, we can use these inducible versions of CRISPR-I to control transcription, and in that way, we can set up experiments and then knock down an essential gene when we want, and then carry out analysis. So here below, this is the same inducible system where before we're looking at knocking down these essential genes over the course of several days. If we don't add doxycycline to turn on expression of the CRISPR-I construct, we can see that these sgRNAs targeting essential genes have no effect on cell proliferation, so the cells proliferate like wild-type cells. If you then knock down these variety of essential genes, you see that the cells where you've knocked down the essential gene deplete very robustly relative to their wild-type counterparts. And then lastly, we've shown that you can use this to control expression of both protein coding genes and non-coding genes. This is a little bit later work than what I showed earlier, where we're knocking down a variety of endogenous genes. And you can see that our ability to control transcription has gotten more robust over time as we've derived algorithms that are more highly enriched for highly active guides. So this is qPCR data on the top and bottom, and it's a log10 plot. So you can see we're really, really robustly controlling transcription for many of these genes, both protein coding and non-coding, where we're getting like 99.9% repression. And if you look by, for example, FISH down here for exist RNA, which codes the inactivated X chromosome, you can see that there is like really no signal left at all for these cells. So we're completely turning off transcription for this endogenous non-coding RNA. So all that work was done in cancer cells, a chronic myeloid leukemia cell line called K562s. Well, not all of it, but most of it. Some was done in HEC293s and HeLAs, but really like standard sort of very robust and easy to use cancer cell lines that may or may not model many types of biology. And so we were curious whether we could expand our ability to control transcription out of cancer cells into more developmental models or other models. And so together with Bruce Conklin's lab, who's in the Gladstone at UCSF, we decided to ask whether we could control transcription in human iPS cells. And so what we did was, rather than all the previous work I showed was either transient transfection or lentiviral delivery. In this case, we've gone to knocking our constructs into a safe harbor locus within the cell, so we can control their expression very stably over time in iPS cells. And then we used the same inducible system that I showed on the previous slides to ask how efficient is our ability to control transcription? Is this tunable? Is it reversible? Can we multiplex this? We continue to ask many of the same questions. And so this is a series of pictures comparing the ability to the gene's not on here. They're targeting sorry. what is this? So this is actually just a schematic to show that the system works, that you can knock these constructs into the genome of an iPS cell, and that we can control expression of either Cas9 or dCas9 within a regulatable system. So in both cases, we've targeted either our CRISPR-I constructs or a Cas9 nuclease to the AAVS1 locus. We can use a TALEN to make a double-strand break, which increases homologous recombination, and then we have use a long double-stranded linear donor to knock in all of this business into these AAVS1 loci. And so what we've knocked in is both a constitutive promoter driving the RTTA that in the presence of doxycycline will activate expression of a CRISPR-I construct or a Cas9 nuclease. And you can see that in the absence of doxycycline, the CRISPR-I construct or Cas9 are not expressed. If you add doxycycline, you get homogeneous expression across the population of cells. So then the question is this functional? And so Bruce's lab targeted a variety of genes, but shown here is targeting NANOG with either CRISPR-I or CRISPR-N, and we're able to show that here pretty good control of gene transcription. If you target NANOG with CRISPR-N, you see more heterogeneity. So some cells will be completely knocked out for NANOG, other cells will be heterozygous and still express some NANOG. Both tools are great. They just give you different outputs. Here you're not making nulls, you're turning transcription down. Here you make true nulls, but you also have heterogeneity in protein expression across the population. And Bruce's lab showed that this system in general, as in cancer models, works quite robustly to control transcription of a variety of types of endogenous genes, such as qPCR data. But overall was a nice proof of principle set of experiments showing that we could turn off transcription in iPS cells in an inducible way. And so you can imagine this is especially important for studying differentiation, where you might want to grow your iPS cells up, infect in a guide, induce some differentiation protocol, for example, that guides you down a neuronal lineage or a cardiac lineage, and then at a specific time point during that differentiation process, turn off a gene and ask how is that gene required for neural differentiation or cardiac differentiation. And so that's all possible with either of these systems, really, in most ways, but definitely we're able to control transcription in iPS cells. One of the things we noticed really early on that I like to re-emphasize in this workshop is that in many settings in human cells, expression of an sgRNA is the limiting factor for a lot of types of experiments, whether it's imaging or in many cases editing, if you're not using RNPs, or for controlling transcription. So one of the ways we saw this was that we were knocking down these GFP reporters, and we saw that the level of knockdown, so this is expression of a GFP reporter, was to some degree correlated with indirect measurements of guide expression. So the more sgRNA you express, the more you repress this GFP reporter. And so that was a problem for us early on, because we had an eye towards doing functional genomics where we perturb expression of all genes encoded in the genome. But to do that, you need to build a platform that relies on guide expression from a lentivirus that's integrated at single copy into the genome. And so if we had a dose-limiting expression of our sgRNA, that wasn't a very satisfying position to start from for building a functional genomics platform. And so together with Bo Huang's lab and Stanley Chi, we tested a series of changes to the original sgRNA design. So this is the original published design from Jennifer that was published early on. It's actually a modified form of the natural Pyogenes hairpin that's a bit truncated. So it's truncated by 5 bases. So we tested a number of things, but we tested just what happens if we just extend the hairpin to the wild type length, and that seemed to be more active, as these were the active changes that we made. And then the other thing we noticed is that there's a Pol3 terminator in the stem of the Pyogenes tracker RNA. And so if we remove that stretch of 4 T's and just break it up with an A, you increase expression of the guide RNA by about 5-fold. which is beneficial in many cases. And then if we combine both this change that stabilizes the sgRNA structure along with the change that increases transcription, what we saw was that we got a lot better activity when we were trying to control transcription with single copy lentiviral integrations of the guide. So this isn't always effective if you're doing transient transfection experiments. Often, expression off of plasmid is really robust, and your experiments won't be limited by either the expression of the Cas9 or dCas9 proteins or the guide. They're just more limited by other factors. But we and others have seen that this guide design in our hands works a lot better. And just so you don't think I'm only talking about our work, this is one example of another lab from Texas, I think, that showed the same thing. And at this point, there are 4 or 5 other papers that have shown that this modified guide design is generally better than what most people are using. So it's a bit, I'll get off my soapbox, but basically it's a bit surprising that people haven't switched toward this, because there's no consequence as far as we can tell to having, well, unless you want an inducible system like Dana's using. But in many cases you want to maximize your guide expression, as these changes help with that. So there are a variety of tools for silencing transcription or doing loss of function experiments, including RNAi and CRISPR, and now CRISPR-I in human cells. And so we were excited about additional functions that we could specify using dCas9. So we tested whether we could activate transcription or as well. You can imagine this would have a variety of applications for gain-of-function experiments that could be useful in studying cancer biology or development. And so, again, we took a page from the artificial transcription factor field and used the sort of who's who of domains to try to activate transcription of reporters. We fused this VP16 domain in 4 copies to dCas9, and then targeted a GFP reporter and asked this reporter importantly has 3 binding sites for the sgRNA we're expressing, and asked whether we could turn GFP expression on. So if you look a couple days later by flow cytometry, we can see that specifically when we co-express the dCas9 activator protein together with a guide targeting this promoter, we turn on GFP expression. So this is proof at least that we can fuse these domains to dCas9 and use them to target the genome and activate transcription of reporters. Disappointingly, when we started to try to test our ability to activate transcription of endogenous genes, we saw very quickly that this scheme doesn't really work very robustly to activate endogenous gene expression. And this is not entirely surprising. Yesterday I showed you a slide from Keith Jung's lab where they were using tail VP64 fusions, and I didn't really walk you through the slide, but essentially what the point of the paper was that one tail VP64 fusion is usually not enough to activate transcription of an endogenous gene. And they saw that with tails you had to have 4 adjacent or 3 or 4 adjacent binding events to get activation of most endogenous genes. And so that's essentially the exact same thing we're seeing here, where if you have 3 adjacent binding events you get nice activation of a gene, where if you have a single binding site and you're only recruiting 1 copy of this VP64 domain, you don't get much activation. And so at that point I think the field in general started to engage with this idea that a simple artificial transcription factor that activates transcription with a single binding event was not going to be enough, or at least not enough with a single activation domain. And so we and others started to search for additional ways to recruit more activation domains with the expression of a single sgRNA. And the way we solved this problem was we fused a highly optimized epitope tag to dCas9. So this is derived from a yeast GCN4 protein. It's a fairly linear epitope that can be connected by a short flexible linker. So we fused 10 copies of this epitope to dCas9. And so now in theory, with expression of 1 sgRNA, if we co-express a single chain variable fragment fused to this same activation domain, we can recruit up to 10 copies of the activation domain, sort of using a single sgRNA with a version 2 system to amplify our signal. And we showed in human cells for a variety of endogenous genes that this really worked much better than the version 1 system. So here we're looking at just a test reporter gene called CXCR4. The gene's not really important. For the aficionados, this is a HIV co-receptor. And you see with our version 1 system, when we target CXCR4, we really don't activate transcription at all. If you use our version 2 system, where we're now recruiting 10 copies of this activation domain, we see we turn on gene expression by about 40-fold. This gene is pretty much off in these cell types, so it's difficult to normalize a fold change. If you subtract background fluorescence, you can get artificially large fold changes, and so field struggled a little bit with how to display these kinds of data. What I like a little better is just to display the primary flow cytometry data. So these are K562 cells that are unstained, so this is autofluorescence. If you stain them for CXCR4 expression and in cells that express a negative control guide RNA, you see, they look the same as the unstained cells, indicating there's really almost no CXCR4 expressed on this cell type. If you then activate CXCR4 expression by expressing a guide in the presence of this version 2 CRISPR-A system, we see that most of the cells in the population activate expression, and it's fairly homogeneous across the population. And so our ability to, a little bit of a subtle point, but sorry. I'll answer the second question first. No, we don't. For the most part, we don't see positional effects where you activate neighboring genes. If you have two anti-parallel promoters that are very close together, you'll activate both of them. But the window in which we activate transcription is quite narrow. It's only about 1 to 2 kb. So the transcription start sites for endogenous genes are generally more spaced out. There are caveats to that answer. There are a lot of described non-coding RNAs that are anti-parallel to coding genes that are very, very close with their divergent transcription start sites. And so we can't really decouple the function of an adjacent non-coding RNA, although that's a bit of a tricky thing to say, because often the expression of those anti-parallel non-coding RNAs is intimately also involved in regulation of the coding gene. So you might need additional methods to dissect those. For the first question, that's sort of hard to measure. The affinity between the epitope that we're using and the scFv is extraordinarily high. I would be surprised if you didn't have the scFv bound to dCas9 constitutively, and that we haven't measured it. If you do, like we've done some FRAP experiments where we're just imaging telomeres using this same setup, and I don't know, that doesn't really tell you the answer to your question. I don't know. So like I said, the field was searching for a series of second-generation CRISPR-A constructs. There have been quite a few published at this point, from us and Ron Vale, from George Church's lab, from John's lab, and Janish's lab, and who else is on here? Charles Gershbach, maybe Stanley. Anyways, there are at least 8 now published. And George Church did the field a service by comparing them all in a Nature Methods paper described here last year. And I have a few issues with the way he did this, but we'll just take him at his word. And he says that ours, this Viper system, and the SAM system from Fung are the best. All three sort of convergently arrived at the same idea that you need to use multiple activation domains to robustly control transcription of, or robustly activate transcription of endogenous genes. And so you can read about these, they're all published, but they all converge on the same idea essentially. So I want to introduce an idea that's been really, really useful over the years in synthetic biology. So far today I've talked predominantly about controlling transcription or doing a variety of things by fusing proteins together artificially. Another way that you can control protein localization is to use RNA aptamers that have high affinity for proteins. So have been used or that have been